You know, in the world of medicine today, we are sitting on an absolute mountain of data. And for diseases like cancer, the clues are hidden right there, inside the genetic code of every single cell. But here's the problem. How do you find that one critical signal, the real smoking gun, when it's buried among thousands and thousands of innocent bystanders? Well, today, that's exactly what we're going to do. We're going to decode cancer's genetic signature. So here's the game plan for this explainer. First, we're going to lay out the challenge. It's like finding a genetic needle in a haystack. Then we'll meet our two expert detectives, two different statistical methods named Sam and Limma, and they each have their own way of hunting for clues. After that, we'll see what they found, their final conclusion, and we'll wrap it all up by looking at the genetic mugshot they help us create. All right, let's dive in. Our whole investigation really starts with this one central mystery. How on earth do you sift through a massive, overwhelming amount of genetic data to find the specific signals that actually define a disease? And that brings us to the fundamental question, right? It's the one that drives this entire field. It's not just about finding any difference. It's about finding a difference that is truly meaningful, one that's statistically solid and biologically important. Our case study is going to focus on 38 patients who have two different but similar types of leukemia. We've got 27 patients with ALL and 11 with AML. Now, to the naked eye, these diseases can look alike, but we know that genetically they have unique fingerprints, and our job is to find them. And here is the scale of our challenge. For each one of those 38 patients, scientists measured the activity of 3,051 different genes. I mean, do the math. That's over 115,000 data points to sort through. This is our genetic haystack. And somewhere, buried inside all that data, is the signature that clearly separates ALL from AML. Okay, so how do we tackle this? Let's bring in our first detective, the Sam method. Its specialty, its real talent, is making absolutely sure that any clue it finds isn't just a fluke, that it's not just random luck. So how does Sam do that? Well, it uses this brilliant technique called permutation testing. Just imagine you're playing cards and you get dealt a perfect, amazing hand. Your first thought might be, was I just unbelievably lucky? To find out, you could shuffle that deck and deal out thousands of random hands, just to see how often that perfect hand shows up by chance. That is exactly what Sam does, but with our patient data. So here's the process, step by step. First, it measures the actual difference in gene activity between the real cancer groups. Then the fun begins. It shuffles the patient labels, randomly calling some ALL patients AML and vice versa, and it recalculates everything. And it does this thousands of times to build a picture of what random noise looks like. Finally, it compares the real result to that random baseline. Any genes that are way, way outside the norm, those are our first suspects. And this plot shows you exactly what Sam found. That straight line going up the middle, that's the line of expectation. It's what you'd expect to see if everything was just random chance. But look at those red dots. Those are the genes that are straying far, far away from that line. They are the ones Sam has flagged as significant. It's incredibly unlikely they'd be there just by accident. Okay, that was detective number one. Now let's meet our second expert, the Limma method. And Limma has a totally different approach. It doesn't look at each gene all by itself. Limma believes there's strength in numbers. Limma uses a technique called the empirical Bayes method. And the idea behind it is just fascinating. Instead of treating each of our 3,051 genes as a separate, lonely little experiment, it looks at all of them together. It learns about the overall patterns across the entire dataset, and then it uses that bigger picture knowledge to make a smarter, more reliable judgment about each individual gene. It literally borrows strength from the whole group to analyze each part. And here we have Limma's result, shown in what's called a volcano plot. This thing is so powerful. Let me break it down. The horizontal axis measures how much a gene's activity changed. The further from the center, the bigger the change. The vertical axis measures how statistically significant that change is. The higher up, the more confident we are it's real. So where are our prime suspects? They're in the top corners. Those are the genes with a huge change in activity that is also highly statistically significant. Okay, so let's recap. We've had two brilliant, independent detectives analyze the exact same evidence using completely different methods. Sam was checking for luck, Limma was borrowing strength. So, what's the verdict? Our first detective, Sam, after running all its shuffles and checks, came back with a list of 131 highly significant genes. And our second detective, Limma, using its strength and numbers approach, identified a list of 129 prime suspects. Now, just look at this. This is the kind of moment that gives scientists goosebumps. The two methods, they found an almost perfect overlap. 129 of the exact same genes showed up on both lists. This is huge. When two completely different ways of looking at the data point to the exact same suspects, you can be incredibly confident that you found a real biological signal, not just some random statistical noise. So now that we have this high-confidence list of genes, we can finally create the last piece of evidence. We can build that visual mugshot that captures the unique genetic identity of each of these cancers. To do this, we use a tool called a heatmap, and what this chart is going to do is take those 129 key genes and use their activity levels to create a unique visual fingerprint for every single one of our 38 patients. This is really the culmination of our entire hunt. Now, it might look a little complicated, but it's actually pretty simple to read. Each vertical column, that's one patient. Each horizontal row, that's one of our key genes. And the color tells you what that gene is doing in that patient. Bright red means it's super overactive, and bright blue means it's super underactive. Simple as that. Now, step back for a second and just look at the big picture with me. Do you see it? The pattern isn't random at all. The data naturally forms two very distinct blocks of color. One of those blocks is almost entirely the ALL patients, and the other block is the AML patients. This picture is the proof. These 129 genes really are a reliable signature that can tell these two cancers apart. So, after all that, you might be thinking, okay, cool, but why does this actually matter? Well, it's a huge deal. Finding a genetic fingerprint like this is revolutionary. For one, it can lead to much more accurate diagnoses. And maybe even more important, by understanding exactly which genes are going haywire, it opens the door for scientists to develop targeted therapies, medicines designed to fix the specific problem driving the disease. You know, we started with this absolute mountain of data and a really complex question. But by using these powerful statistical tools, we managed to cut right through the noise and find a clear, actionable signal. And that really leaves us with a final, exciting thought. If we can see a cancer's unique genetic signature this clearly, what's next? What other diseases are we going to decode?